Biotechnology is a rapidly advancing field that harnesses biological systems and organisms to develop innovative technologies for various applications. In the realm of biotechnologies, two areas that hold immense promise for the future are cloning and artificial organs. Cloning, a technique that involves creating genetically identical copies of organisms, has revolutionized the way we understand genetics and reproduction. One of the most famous examples of cloning is Dolly the sheep, the first mammal to be cloned from an adult somatic cell. This breakthrough opened up possibilities for medical research, agriculture, and conservation efforts. Cloning techniques have since evolved, allowing scientists to clone specific genes, cells, and even organs. The applications of cloning are vast and diverse, ranging from producing genetically modified organisms for agriculture to creating animal models for studying diseases. However, the ethical considerations surrounding cloning, particularly human cloning, remain a topic of debate and scrutiny. On the other hand, the development of artificial organs is another exciting frontier in biotechnology. With advancements in tissue engineering and regenerative medicine, scientists are now able to create functional artificial organs that can potentially replace damaged or failing organs in the human body. Artificial organs offer hope to patients awaiting organ transplants and could alleviate the shortage of donor organs. Despite the remarkable progress in artificial organ development, challenges such as biocompatibility, long-term functionality, and cost-effectiveness persist. Looking ahead, the future of biotechnologies holds great promise for further advancements in cloning techniques and artificial organ development, paving the way for groundbreaking innovations in healthcare, agriculture, and beyond. Cloning techniques have come a long way since the groundbreaking success of Dolly the sheep. One of the most common methods of cloning is somatic cell nuclear transfer, T, which involves transferring the nucleus of a somatic cell into an egg cell that has had its nucleus removed. This process reprograms the somatic cell to become a pluripotent cell capable of developing into a new organism. Eskin has been used to clone various animals, including mice, cows, and even monkeys. Another cloning technique is embryo splitting, where an early-stage embryo is divided into multiple parts, each of which can develop into a separate organism. This method has been used to create genetically identical twins in animals. In recent years, scientists have also explored the use of induced pluripotent stem cells, iPSCs, for cloning. iPSCs are adult cells that have been reprogrammed to behave like embryonic stem cells, offering a potential alternative to traditional cloning methods. Interestingly, there have been attempts to clone extinct species using preserved DNA samples. For instance, the Pyrenean ibex, a species that went extinct in 2000, was briefly brought back to life through cloning before succumbing to health complications. Cloning techniques have not been limited to animals. Plants have also been successfully cloned using methods such as tissue culture and plant grafting. These techniques have enabled the mass production of genetically identical plants with desirable traits, revolutionizing agriculture and horticulture. Despite the advancements in cloning techniques, challenges remain. One major hurdle is the low success rate of cloning, with many cloned embryos failing to develop or resulting in abnormalities. Additionally, the ethical implications of cloning, particularly human cloning, raise complex questions about identity, individuality, and the sanctity of life. As technology continues to advance, researchers are exploring new avenues for improving cloning techniques, such as gene editing tools like CRISPR-Cas9. These tools offer the potential to precisely modify genes in cloned organisms, opening up new possibilities for genetic engineering and personalized medicine. The future of cloning techniques holds great promise for advancing our understanding of genetics, reproduction, and the potential applications of cloning in various fields. Cloning, once considered a realm of science fiction, has found numerous practical applications across various fields. One of the most significant applications of cloning is in agriculture. Farmers can use cloning techniques to produce genetically identical plants with desirable traits, such as disease resistance or high yield. This has revolutionized crop production and helped ensure food security in many parts of the world. In animal husbandry, cloning has been used to preserve valuable genetic traits in livestock. For example, prized dairy cows or racehorses can be cloned to produce offspring with the same superior qualities. Cloning also plays a crucial role in conservation efforts. Endangered species with limited genetic diversity can benefit from cloning to increase their population numbers and genetic variability. The successful cloning of endangered animals like the Javan banteng and the African wildcat has offered hope for the preservation of these species. In the field of medicine, cloning has opened up new possibilities for regenerative medicine and personalized treatments. Scientists can create animal models of human diseases through cloning, allowing for the study of disease mechanisms and the development of potential therapies. Cloning has also been instrumental in producing stem cells for research and therapeutic purposes. Induced pluripotent stem cells, iPSCs, generated through cloning techniques hold promise for regenerative medicine and the treatment of various conditions, such as spinal cord injuries and heart disease. Beyond agriculture and medicine, cloning has found applications in biotechnology and industry. Biopharmaceutical companies use cloning to produce large quantities of proteins, enzymes, and antibodies for therapeutic purposes. Cloning has also been employed in the production of biofuels and biodegradable plastics, contributing to sustainable practices in industry. The versatility of cloning techniques continues to inspire innovation and drive progress in diverse fields, from agriculture to healthcare to environmental conservation. Cloning technology presents a myriad of ethical considerations that have sparked intense debate and scrutiny. One of the primary ethical concerns surrounding cloning is the potential for abuse and exploitation. The ability to create genetically identical copies of organisms raises questions about individuality, identity, and the sanctity of life. In the case of human cloning, there are fears of reproductive cloning being used for unethical purposes, such as creating designer babies or cloning individuals without their consent. Another ethical dilemma is the welfare of cloned animals. Cloning can result in high rates of miscarriages, stillbirths, and health issues in cloned animals, raising concerns about animal welfare and suffering. Additionally, the commercialization of cloning raises ethical questions about the ownership and patenting of life forms. Companies that clone animals for profit may prioritize economic gain over animal welfare and conservation efforts. Furthermore, the long-term implications of cloning on genetic diversity and ecosystem stability are subjects of ethical concern. Cloning endangered species, for instance, may offer short-term solutions but could have unforeseen consequences on natural ecosystems. Despite the potential benefits of cloning, ethical considerations must be carefully weighed to ensure that the technology is used responsibly and ethically. The development of artificial organs represents a groundbreaking frontier in biotechnology, offering hope to patients with organ failure and revolutionizing the field of regenerative medicine. Artificial organs are created using a combination of tissue engineering, biomaterials, and regenerative techniques to mimic the structure and function of natural organs. One of the most well-known artificial organs is the artificial heart, which has been developed to replace damaged or failing hearts in patients awaiting heart transplants. These devices, such as the total artificial heart, TA, are designed to pump blood throughout the body and sustain life while patients await a donor heart. Another remarkable advancement in artificial organs is the development of bioengineered kidneys. Scientists have successfully created functional kidney tissue using a combination of stem cells, biomaterial scaffolds, and 3D printing technology. These bioengineered kidneys have the potential to revolutionize the treatment of kidney disease and alleviate the shortage of donor kidneys for transplantation. Artificial lungs are also being developed to support patients with respiratory failure. Lung assist devices, such as the Hemolung Respiratory Assist System, provide temporary respiratory support to patients with acute lung injury or chronic obstructive pulmonary disease. In the realm of artificial pancreas development, researchers are working on creating implantable devices that can regulate blood sugar levels in patients with diabetes. These artificial pancreases combine glucose sensors, insulin pumps, and control algorithms to mimic the function of the natural pancreas. Interestingly, advancements in artificial skin development have led to the creation of skin substitutes that can promote wound healing and tissue regeneration. These bioengineered skin grafts are used in the treatment of burns, chronic wounds, and skin disorders. The field of artificial organs continues to evolve rapidly, with ongoing research focused on developing more complex and functional artificial organs, such as the artificial liver and artificial cornea. These advancements hold the potential to transform healthcare by providing innovative solutions for organ replacement and regenerative therapies. Artificial organs offer a multitude of benefits that have the potential to transform healthcare and improve the quality of life for patients. One of the key advantages of artificial organs is the ability to provide life-saving treatments to patients with organ failure who may not have access to donor organs. These devices serve as temporary solutions while patients await organ transplants, reducing the risk of mortality and improving patient outcomes. Additionally, artificial organs can alleviate the shortage of donor organs, which is a significant challenge in organ transplantation. By providing alternative options for organ replacement, artificial organs offer hope to patients on transplant waiting lists. Another benefit of artificial organs is the potential for personalized medicine. These devices can be tailored to individual patient needs, offering customized treatment options based on specific medical conditions and requirements. Furthermore, artificial organs reduce the risk of organ rejection, a common complication in organ transplantation. Because artificial organs are made from biocompatible materials, the risk of rejection by the immune system is minimized, leading to better long-term outcomes for patients. Overall, the development of artificial organs represents a major advancement in medical technology, offering innovative solutions for organ replacement and regenerative therapies. The development of artificial organs presents a range of challenges that must be addressed to ensure their effectiveness and safety in clinical applications. One of the primary challenges in artificial organ development is achieving biocompatibility. The materials used to create artificial organs must be compatible with the human body to prevent immune reactions and rejection. Ensuring that the artificial organ integrates seamlessly with the surrounding tissues and functions properly is crucial for long-term success. Additionally, maintaining the functionality and durability of artificial organs poses a significant challenge. Many artificial organs, such as artificial hearts or kidneys, must perform complex physiological functions continuously without failure. Designing these devices to withstand the rigors of daily use and mimic the intricate functions of natural organs is a formidable task. Another challenge in artificial organ development is the issue of vascularization. Natural organs are highly vascularized, with intricate networks of blood vessels that supply oxygen and nutrients to cells. Replicating this level of vascularization in artificial organs is essential for their survival and function. Without proper blood supply, artificial organs may suffer from ischemia and necrosis, limiting their effectiveness. Moreover, scalability and mass production present challenges in the widespread adoption of artificial organs. Creating artificial organs on a large scale to meet the demand for organ replacement therapies requires efficient manufacturing processes and quality control measures. The cost-effectiveness of artificial organs is also a significant hurdle. Developing affordable, artificial organs that are accessible to a wide range of patients is essential for ensuring equitable healthcare. Regulatory approval and ethical considerations add another layer of complexity to the development and deployment of artificial organs. Ensuring that artificial organs meet stringent safety and efficacy standards set by regulatory agencies is crucial for their clinical use. Addressing ethical concerns related to the creation and use of artificial organs, such as patient consent and privacy, is essential for maintaining public trust and acceptance. Despite these challenges, ongoing research and technological advancements continue to push the boundaries of artificial organ development, offering hope for improved treatments and outcomes for patients with organ failure. The future of biotechnologies holds immense promise for groundbreaking advancements that could revolutionize healthcare, agriculture, and environmental conservation. One exciting prospect is the development of personalized medicine through the use of genetic testing and targeted therapies. By analyzing an individual's genetic makeup, healthcare providers can tailor treatments to specific genetic markers, improving treatment outcomes and reducing adverse effects. Another fascinating area of research is the use of gene editing technologies, such as CRISPR-Cas9, to modify genes and potentially cure genetic diseases. CRISPR-Cas9 has already shown great potential in correcting genetic mutations in laboratory settings, offering hope for treating conditions like sickle cell anemia and cystic fibrosis. In agriculture, biotechnologies hold the promise of enhancing crop yields, improving food security, and reducing the environmental impact of farming practices. Genetic engineering techniques can be used to develop crops that are resistant to pests, diseases, and environmental stressors, ensuring sustainable food production in the face of climate change. Furthermore, biotechnologies offer exciting prospects for environmental conservation, such as the use of bioremediation to clean up polluted environments and the development of biofuels to reduce reliance on fossil fuels. By harnessing the power of biotechnologies, we have the potential to address some of the most pressing challenges facing our world today and pave the way for a healthier, more sustainable future.